This is how. Hello, this is a video to train you on the basis of the biology classes. So DNA, when we start doing DNA, we do not really use microscopes. So when we start talking about cells, it would be impossible to do that without it. Now, this is the kind of microscopes we have for most classes. The advantage is that this doesn't need a computer, so we do not have to worry about the school's permission. When you explain to your students how to use it, even if they've got it at home, remember, the first thing you need to tell them is that there are two factors, only two factors. Magnification, how big you make it. And here it's quite clear. You see 200 to 60. And this is, let me say this is something for both juniors and seniors, although seniors will use it much more. So they both have to understand there are two things that you have to worry about when you work with your microscope. It's magnification, so how big do you want your vision to be, and focus. And if they've got cameras, they understand what focus is. So there's no hard and fast rule. Any and each of your juniors and seniors, although for the juniors they need a lot of help, they will have to literally bend down. So switch on the light. I don't know if you can see it, but the light is on. Bend down over the slide. And then really try and work on the focus and on the magnitude. So for example, the first thing you could do before you introduced slides and covers is ideally there will be one of these per each team. So for example, one member of the team can offer their skin and the others just work on the zoom and on the magnification to see, can they see it? Once everybody's done this, or I can see, oh, this is my right focus, or this is my right magnitude, then you introduce the next thing. And this is for the next video. So this is really the basics, both for juniors and seniors, how to use a microscope. Magnification, so how big you want this to be. The image, how much bigger do you want the image to be. And focus. That focus really speaks for itself. Don't forget to switch on the lights and off. On, off. And that's about it. Hello, and this is another training about how to use the microscope once the children have become used to it. As you can see now, we have got a microscope. Underneath, we've got a slide. This is a pre-prepared slide, and this is something that both juniors and senior will use. But at first, ironically, it will be the juniors, because you do not have time. You do not have the time, and they haven't got the capacity to make their own. So first of all, show them that there's a list saying what slide the slide represents. Another important thing is that if you do not manage, if you have chosen, for example, and this is what I always say, start with the same things that the seniors do. So if the seniors are doing they're choosing to check the cells in an onion peel, we have got this too, and it's number 24. Now, I sometimes find when the whole set is ready, I find pulling out slides a bit difficult. So if you can, pull the one before, pull the one after, and gently pull yourself one. When I say yourself, I mean the teacher or the assistant, not the children, because they are very fragile. Once you've got them, place them under. I really recommend that you get yourself some white paper underneath, because it really makes it easier. And start teaching them the proper technique. And it's not easy. It's taken me about 5 minutes, literally, just to bend over and find the right magnitude and the right focus. And now I can see actually the section of an onion in a beautiful blue. I can see the focus, I can see the wall. It really is very clear. But do not assume that your students will just find the right focus and the right magnitude the moment they bend down. Be prepared to help them, whether they're juniors or seniors. Obviously, do more for the juniors. And this is yet another video to talk about preparation for and with your microscope. So if you're watching this, you've watched both number 1 and number 2. This is number 3. So you know that this is why we use this one. It's the most school friendly. You do not have to bring a laptop with you. But now, let's say you have already sorted out the juniors, they are doing their pre-prepared, they've all checked their pre-prepared slides. You, on the other hand, you and your seniors are preparing yourself the section. So if you're watching this, you already watched the video that we have uploaded about how to check the cells in an onion. And this is just in preparation of. Always have white paper. I really recommend it. Make sure your students know how to use a slide. Clean, nice slide. And get them, before you start, get them to practice on how to put the cover on, making sure that there's not a lot of air and dust on it. Now, I don't know about you, but I am not very dexterous, so I'm a bit of a disaster. So this is my coping technique. I use this, I don't know, this little stick. It could be also a little bigger. Make sure they're definitely wearing goggles while they're using this. You do not want anyone being poked in the eye. Then keep the sticky part of, the pointy part of the, whatever you use, whatever stick you've used, holding on to the little cover, and then gently, very, very gently, very, very gently, very, very gently, very gently, make sure that the cover is on top of whatever. Let's say that here, for example, right in the middle, you've got your little section of skin. So the skin has been stained, the staining agent is already here. You want to make sure there is no air bubble, and this is why you've done this. Let's do this again. You have moved with the pipette, whatever, actually you have moved with something, whatever sample you're checking. In this case, for example, it should be a section of onion peel. So the onion peel stain is here. Now you want to cover it, nice and, now you want to cover it, nice and gentle, nice and gentle, nice and gentle, nice and gentle. Now you've got the cover on top, and you must make sure that there is no air bubble and there's no dirt. Once you've done it, again, you take your microscope, and again, you get them to bend down and to find the right focus and the right magnitude. It seems simple and it's not difficult, but it really requires a lot of patience. Make sure that you've got one per each group. Thank you so much.
Created using Powtoon.